My PhD work was at Warwick University and the Natural History Museum in London, and was together with Robin Ellaby and Mark Kareen. To give you a little bit of an idea what Trochetiopsis looks like, you have here an image on the right. They used to be trees of about 2-3 meters tall. I think on the island today, there are some that are growing this tall, but they're still, a little bit more shrub-like, most of them. And to really start my story about my PhD work, we have to go back a little bit further in time to James Cook's first voyages. And these voyages, or he made 3 voyages, and during these voyages, he had naturalists on his ship that would explore the places that they went to. And they would collect flora and fauna from these very remote places and bring them back to England to study them further. And you can see here that they passed by St. Helena on every occasion, and especially on their first voyage on their way back, which is the Green Line. In 1771, they made some very remarkable and important collections. So these travels have been very key to my research. I don't have to tell you much about St. Helena, I think, but I do want to emphasize that St. Helena is relatively small for a volcanic island, and has also been one of the reasons that it was so interesting for us as a study area. And of course, it has the interesting history of also human impact. So it was discovered in 1502 by the Portuguese, and they put goats on the island, but they did not colonize it. Colonization happened by the English in 1658, and by that time they did get rid of quite a few of the goats, as they had been roaming around freely. The goats' population became really big, and they did affect quite a bit of the island vegetation. Also, the English, when they colonized the island, introduced several non-native species, tree species, and crops, which also, again, affected the island. So only 3.5% of the island area contains endemic plants. And from all of the endemic flora, we lost about 30%, which is a shame. But here today, we focus on one of the genera, which is Trochetiopsis. And Trochetiopsis consists of 3 species. On the left, you see Trochetiopsis melanoxylon, which is also referred to as the St. Helena dwarf ebony. Ebony because of the dark wood that it has, or had. And it has been extinct and was last collected in 1771. So that is during that James Cook's first voyage. And we think it went extinct around 1780, but we're not entirely sure. Then there is Trochetiopsis erythroxylon, which is the St. Helena redwood, again referring to the redness of the wood. And this is extinct in the wild since 1968, but we can still find it in cultivation on the island, but also in the UK. And then we have Trochetiopsis ebonus, which is the St. Helena ebony. And this was rediscovered in the 1980s. It was thought to be extinct before this time for about 100 years. And after its rediscovery, it was reintroduced to the island. So it's now present on the island in several places, but also still in cultivation in the UK and also in other places like the Netherlands and across Europe. Yes, next slide. So then a little bit more about the history of Trochetiopsis. We, of course, in 1500s had the goats that had a massive effect on the population decline of this group. And we have the English colonization. We have the first collection of Trochetiopsis, which was made in 1698. And then we had a very remarkable notion of one of the naturalists, Banks, in 1771, that said that the trees that produced it, which was the dark ebony wood, are now nearly extinct, and no one remembers the time when they were at all plentiful. Which is very interesting. So it really is a remark on, we know that these populations were very big and the trees were across the entire island, but already around the end of the 1700s, they couldn't really remember that time, and the trees population already declined a lot. Later on, in 1843, we have Hooker, who mentioned during his lecture that probably 100 St. Helena plants have thus disappeared from the Systema Naturae since the first introduction of goats on the island, which is one of, I think, one of the first remarks on human impact on the island, which is very interesting as we kind of know about it, but really people already saying it at that time was quite important. Unfortunately, it did not quite help to save some of the tree species. And yeah, later on, Erythroxylon went extinct. But of course, we have the rediscovery of Trochetiopsis ebonus in the 1980s. Okay, then we have why the other things that make Trochetiopsis very interesting for us to study. Since Trochetiopsis is sort of an adaptive radiation. So we have speciation of these 3 species into their own habitat, which is has happened in this very relatively small island. And we see that Melanoxylon occurred mainly in the north near Jamestown, that Erythroxylon was more in the middle of the island in the higher areas around Diana's Peak, and then Trochetiopsis ebonus in green was more in the southwest. We have historical records of the human impact and what happened with the populations over time, and we have historical collections that span the 320 years. So my research motivations were really about how did this anthropogenic change impact the island diversity patterns, specifically for Trochetiopsis. We know it was a big population at one time. Now we know it's a relatively small population. So how did that change actually happen more on a genetic level? And if we look on this genetic level, can herbarium collections help add more genetic information than if we only look at the modern samples that we can find on the island and in cultivation today? What can we discover from the past 3 centuries about Trochetiopsis genetic change? So did the genetic diversity decline, or did something else happen, or was it too short of a time to really notice any change genetically? So, yes. So I've sampled several herbaria collections, in total 14. And I sampled 3 modern specimens from Chelsea Physic Garden. Here you can see on the left, the y-axis is the years, so the collection years that were made. And then you see on the x-axis Trochetiopsis ebonus in green, Trochetiopsis erythroxylon, and so on. Ebonus, we have a gap of 100 years. This was when it was thought to be extinct. So we have tried to have one of the older collections somewhere in the middle and then a modern collection. For Erythroxylon, we were able to sample across time. And Melanoxylon, we have a very early collection and the last collection that was made of this species. And then we also have Trochetiopsis benjaminii, which is the hybrid. And I do want to emphasize that we collected not everything at the same time, but we started with a modern material, adjusted our lab protocol so we would know it would work, then went to collections that were a little bit older and again checked our lab protocol. And this was a very nice process. So we could actually say for certainty that when we were sampling the really old historical valuable material that we would be able to get DNA from it. So these old collections are held in the Sloane Herbarium, which is part of the Naturalistic Museum of London. And doing genomic research on museum collections or on herbarium collections, we call museomics or herbariomics. And this is, we refer to this differently because it has fragmented DNA. The DNA can fragment very quickly if you dry a specimen. So it doesn't matter necessarily how old something is, but we very quickly talk about ancient DNA when the DNA is fragmented. And here I have indicated 2 chemical processes that happen with water. You have on the left deamination, where the cytosine base pair loses its NH2 group and then changes into the base pair uracil. And depyrination on the right, where you have guanine, which is a base pair that with water drops off entirely, and then you're left with a depyrinated sugar. Sort of leaving this empty gap can cause friction in the DNA strand and causing a nick. If this happens often enough, you will see that the DNA fragments entirely. So these 2 processes make it a little bit different to analyze and sequence, well, to obtain the DNA, but also to analyze the DNA for this type of research.
Annabelle, so, about 5 minutes left.
Yes. So for the DNA lab work, we really went from getting a reference genome, looking at the fragment, we needed a reference genome for this fragmented DNA to really get the ancient DNA. So we got long reads, and we got this from the Trochetiopsis ebonus, which is the ebonus that you saw earlier. And we created the de novo genome assembly. For this, we obtained PacBio hi-fi sequences, and here we have, I also put the genome estimations, and our assembly was 474 mega base pairs. So our findings, here I have a PCA, where we really look at the differences on genotype likelihood between the different samples. And you can see that the mother material here shows that we have Ebenus and Erythroxylon separate in green and purple as we expected, but the Benjaminii, which was the hybrid, is more closely to Erythroxylon than to Ebenus. Then if we place the more herbarium material in it, the historical material, we see that Melanoxylon in blue is all the way on the left, which is very interesting. So it is definitely a separate species. And we see that Ebenus and Erythroxylon are in vertical lines also separated, and the x-axis PC1 is explaining most, so that makes sense. And in Erythroxylon in purple, we see sort of a time gradient, which is very interesting, with the highest group in the top right from 1884 to 1970. We also did a mixture analysis, which you can see here. And there you can see that the mother material of Ebenus is actually mixed, which was something we did not expect. But then Erythroxylon and Benjaminii grouped together again, and Benjaminii, which was a hybrid and we expected to be mixed, is not, which caused a bit of confusion. If we look again with the older material, we can see Melanoxylon on the left in purple grouping together as expected, Ebenus in red grouping together as expected, and for Erythroxylon, you can see that there is a sort of a time gradient. And we have two groups. So we have a yellow group and a brown group within Erythroxylon, and then the modern in orange. And these groups we thought were kind of over time changes in the population, but we now think that it might be more to do with space, so that these were different populations across St. Helena. One of them being at Diana's Peak, as described in records, and the other groups we are not completely certain where they were from, but we would be able to relate this with other collections at some point. So genetic diversity loss over time, you can see that here. We used Waterstone's estimators, which really looks at heterozygosity, and we can see that for the entire genus, we do see a decline. So there has definitely been a genetic effect on the population that we were able to see, even though it was a very short amount of time in which this happened. So we really think that it's important that if we study islands today, that we don't only look at the mother material, because they only have a fraction of the variation of the herbarium samples. So that it's really important that museum collections reveal a steady decline of diversity due to human impact. So what does that mean for Trochetiopsis? That, well, the cultivated plants are a little bit more mixed than we expected. There is genetic diversity loss, and this does have interesting notions for conservation in the future. Unfortunately, my PhD stopped here, but we are still collaborating with people to look into the conservation, and we will have more genetic information for these future studies. Thank you so much.